Hello folks, we made it to our final macromolecule, good old nucleic acids. Now nucleic acids are still going to be made with carbon, hydrogen, and oxygen. We also include nitrogen and phosphorus. Now when we're thinking of monomers of nucleic acids, we're thinking of nucleotides. So again, because they're nucleic acids, we do have specific names. And of course, a polymer of a nucleic acid is going to be polynucleotides, or you can just call them nucleic acids. And the two major classes that we focus on is deoxyribonucleic acid and ribonucleic acid. The main difference between the two is that we have two different types of sugar. We have a deoxyribose sugar, and then we have a ribose sugar. That's going to help change the structure. We also have different nitrogen bases for them. So let's go ahead and talk about the monomers really quick. So again, I'm going to be drawing my circle. And we know this is definitely going to be a sugar. And it's going to have a phosphate group. And I'm just putting P for phosphorus. And then it's also going to have a nitrogen base. And I'm just going to put NB. And these nitrogen bases are different for DNA and RNA. So for DNA, it's going to be adenine. Okay. It's going to be thymine, guanine, and cytosine. And then for our RNA, they're going to have a difference by the thymine here. So it's going to be adenosine, uracil, guanine, and cytosine for RNA. Okay, another huge difference between them is going to be that DNA is going to be a double helix, whereas RNA is going to be a single strand of nucleic acid. So let's focus on DNA first. Well, the main thing you need to know about DNA is that genes encode protein. You might be thinking, where do all these proteins come from? How are they all being made with specific structures and functions? It all comes from our DNA. It's going to have the genetic blueprint for all of the types of synthesis of proteins we have in the body. So that's a lot of information that is holding. It's also going to be that double helix. So remember we were talking about alpha helices, and when it comes to secondary structure for proteins, well, for nucleic acid, we have one helix, and then we're also going to have another helix as well. It doesn't look perfect, like, it doesn't look that great, but this is what it looks like here. Okay? So mine's obviously is one. Let me go ahead and erase that. And this double helix, again, is going to be made up, trickled down with a bunch of monomers. So let's look at all the monomers here. We have a monomer here, 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 and here. All these monomers down one strand. So if you have one single strand of DNA, you have a monomer, 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 monomer. And the monomers are going to be different based on the different nitrogen bases. So you can end up having like A, T, G, and C for DNA. Okay, that's what the goal is. And then you have another strand over here. And they're also going to have monomers attached to them, but their monomers are going to be based on whatever they are next to. Okay, so what does that mean? DNA has complementary base pairing rules. So these nitrogen bases that are attached to A, T, G, and C, they're always paired with a specific other nitrogen base. So A is always going to be paired with T. T is always going to be paired with A. G is always going to be paired with C. And C is always going to be paired with G. And that's how it goes when it comes to complementary base pairing for DNA. Okay? And the beauty of it is, yes, we are going back to good old hydrogen bonding, folks. Between each of these are going to be hydrogen bonds. And those hydrogen bonds are created because of the different twists and turns that you see in the helix because of those hydrogen bonds as well. So let's look at adenine and thymine here. If I'm looking at adenine and thymine, we see that we can have a hydrogen bond between hydrogen and oxygen here. We're also going to have a hydrogen bond between hydrogen and nitrogen here as well. So because of that, that's what's creating this kind of helix effects on our DNA. All right, folks? So just keep that in mind. And when we're thinking of genes, and you're like, oh, genes encode proteins, we have about over 25,000 genes. And each of those genes can encode different proteins. And so that's where we get all of our information from. So just keep that in mind as well. Let's go ahead and look into RNA. So RNA and DNA, both nucleic acids, right? But RNA is very interesting because there are different types of RNA. And what they do is the DNA is going to have all the genes that's going to encode all the information, and the RNA is going to get that message out and help produce those polypeptides, and therefore all the different level of structure for proteins, which is pretty cool. So RNA is going to be one strand. Again, you're going to have monomers attached. And again, A, U, G, and C. So just keep that in mind. And that addition of uracil, which makes RNA a little bit different than, of course, DNA as well. Okay, folks? And there's different types of RNA. You have messenger RNA, which is mRNA, transfer RNA, and ribosomal RNA. All these RNAs are used to help DNA push out all the orders. So DNA is like, hey, I want to make this protein because the body needs it. So what's going to happen is I'm going to send this message out from the nucleus in the cell, which we're going to talk about later on. And then I'm going to use messenger RNA to send that message out. And then I'm going to use transfer RNA and ribosomal RNA to build the protein that's necessary. Once the protein is built and it goes into the Golgi apparatus, it can get packaged and have all the accoutrements added to it. And then it'll be able to be transported in the cell or in the body or wherever it needs. And that's the beauty of this RNA DNA link. They're both necessary to help protein synthesis be carried out. The DNA is like the CEO, RNA we work with. So just keep that in mind. Okay? Now, if you want to kind of, I have this little chart here that kind of gives the characteristic changes between DNA and RNA. First, DNA is in the nucleus of the cell. RNA is in the cytoplasm. So it's going to be outside the cell. And the mRNA is able to go out of the actual nucleus area. DNA, again, it codes all the genetic codes for protein synthesis. RNA is going to carry out the genetic instructions to build the protein. DNA is double helix. RNA is a single strand. The DNA has a particular sugar called deoxyribose. RNA has ribose. And then the nitrogen bases that are part of the monomers. Remember, the monomer is going to have a sugar. It's going to have a phosphate group. And it's also going to have that nitrogenous base, which could be adenine, thymine, or uracil, guanine, and cytosine. Okay? And so those bases differ for those as well. So when you're going back to our good old table, when we're looking at nucleic acid, they say, what are the elements? Yes, you can say you have carbon, hydrogen, oxygen, nitrogen, and phosphorus. You can keep that in mind. Some people like to say chomp. You know how you can think of chomp, but they say chanop. That's one thing I've seen. Folks who put chon here, chons here as well, and then cho here. So it just depends on how you want to remember it. But the building blocks are going to be nucleotides, and the polymers are going to be nucleic acids. You could call them polynucleotides as well. So just keep that in mind. And if you have two nucleic acids like bonded to one another, on like a strand, for example, then they do have a specific type of bond. You know how proteins have the peptide bond. This has a phosphodiester bond. Extra information, but if you wanted to know what kind of bond, there you go. Okay. Again, the function is figure out, basically genes encode proteins, and you can also just say another function will be to actually carry out the instructions, like RNA carrying out the instructions to produce the proteins or build the proteins or synthesize the proteins. And you can even add more details about RNA, mRNA, tRNA, or transfer RNA as well. It's really up to you. But that's really what the last little bit of this lecture is going to be. Now, finally, you do have a worksheet that you can work on. Some folks want to work on it, some folks don't. I mean, you have the active reading guides you can be looking into. You have the worksheets you can be looking into. For this worksheet, the first thing you're going to do is kind of go through a little bit of details we talked about when it comes to periodic table. Then you have a bunch of terms here, and you're going to organize them in different categories. So if I have ATP, is this ATP going to be a monomer? Is it going to be a nucleic acid, a functional group, whatever it is? So nucleic acid is what it will be, for example. And then we also have something called functional groups. And functional groups are just there as little groups of molecules that then have the ability to go forward and create chemical reactions. So some functional groups would be like OH or hydroxyl. Some other functional groups would be a phosphate group. Okay, so phosphate group. I'm just looking at the thing here. I'm trying to see what other functional groups I can see. Let's see, phosphate amino group. That's another functional group. Again, things that actually have the ability to form, different types of chemical reactions. As we know, an amino group, because they have nitrogen, it can then go forward and do hydrogen bonds as well, similar to hydroxyl here. Carboxyl group, that's another thing that we saw attached to an amino acid. So again, because it has oxygen and hydrogens, it can also form, it can be polar, it can have different hydrogen bonds that it can form as well. So I'm looking at all these here: hydroxyl, carboxyl, amino, and phosphate. And these are the main functional groups that we see that are just involved in a lot of different structures for macromolecules and in how they make reactions. I'm going to erase the phosphate group and redo it because I want to show the phosphate group that's best for our class. So we're going to do that. Okay. Okay. So that's going to be a phosphate group for us. And we've seen these phosphate groups. I'll go back a little bit and show you the phosphate group that we saw here. So you see that little phosphate group there? That's the phosphate group. We have a nitrogen group here. Again, an amino group. We go back to our proteins. We also have an amino group here. We have a carboxyl group here. Okay. We also have an OH that's attached to that carboxyl group. Just trying to go and see all the different things that we have. I know in our carbohydrates as well, and definitely in our phospholipids, we have OH groups as well. Okay. So we'll check everything just to make sure. Oh, here's the go. Example. So we have glucose and fructose, which definitely see an OH group. And then as we go along, we definitely see an OH groups as well. So that's what they mean. That's what they mean. Little the piece by here for functional groups. It's just the groups that can be added to things that maybe are non-polar that then can become polar in some regions. Okay, and that polarity in some regions will allow it to be more reactive. All right, folks. So that is chapter 2. I swear I will be trying to talk less for chapter 3 and as we go on, but yeah, I'll try my best because I know I'll get you talking. Have a great one.